I hope everybody's doing okay this morning. So this morning we're going to cover chapter 19 on proteins, and we're also going to do part of chapter 20 on enzymes. The screen that you see shows a protein. Actually, in the bottom right-hand corner of your screen is a representation of a protein molecule. So that's what we're going to review this morning. The plan is, again, chapter 19 and most of chapter 20 today. Next Thursday at 9 o'clock will be a third exam on chapters 17, 18, 19, and 20. I've given you most of the homework for chapters 17 and 18. I'll do homework problems in chapters 19 and 20, and any other homework problems that you ask me to do, I'll present during the week so that you're prepared for the third exam. So beginning with the protein chapter, then the first topic is amino acids, because amino acids are the building blocks of proteins. Combining amino acids gives you a protein molecule. So there's 20 amino acids. You do not have to memorize the amino acids. If you need amino acid structures, I'll give them to you on the exam. They're called alpha amino acids because the amino group, in this case, looking at the screen, it's the NH3 positive charge on the nitrogen. That's on the carbon that's alpha to the carboxylic group. So were there to be another carbon next to that carbon, it'd be the beta carbon. Alpha is A in the Greek alphabet. So that's why it's called alpha amino acids, because the amino group is on that alpha carbon. What's significant here, looking at this structure, is the R group. So the R group will change. That's why there's 20 amino acids. Each amino acid has a different substituent in place of the R. You'll see that as we go forward this morning. The carboxylic group doesn't have an acidic hydrogen on it. That acidic hydrogen is on the nitrogen. That's why the nitrogen is positively charged and the carboxylic group is negatively charged. So we categorize the amino acids based on their R groups into 4 categories. And you have to know the 4 categories. You don't have to, again, memorize the amino acids, but you have to know the 4 categories. So the first one is neutral and nonpolar. So we're going to go back and forth on these screens to see these. So there's 8 of the 20 have R groups that are neutral, that is, they don't have a charge on them, and they're nonpolar, that is, they're mostly carbons and hydrogens. The simplest amino acid is glycine. It's in the upper left-hand box that you see. It has only a hydrogen on that alpha carbon. It has actually 2 hydrogens, but the R group, the hydrogen represents the R group in glycine. Alanine has a methyl group in place of the R. Valine has an isopropyl group. Leucine has a 4-carbon chain. It's an isobutyl chain, and so forth. So again, you don't have to memorize these, but you have to recognize these as a nonpolar, a neutral side chain. So then let's go back to the list. So next, number 2 is neutral and polar. So these are side chains that are not charged, that's why they're neutral, but they have polar molecules in the side chain. And so a common one is cysteine. You'll note there's a sulfur there, that's the polar molecule. Glutamine and asparagine have nitrogens and oxygens. They're actually amide groups. Again, not charged, but because of these polar molecules, then they're called polar neutral side chains. So you see them here. There's 7 of those. Again, you'll note the oxygens and nitrogens and sulfur polar molecules in the side chain. Going back then third type of side chain is basic. It's got a nitrogen in it, but the nitrogen is positively charged. Unlike the nitrogens that you see below on this screen that are emit nitrogens and not charged, these are amino nitrogens, and they're all charged. So let's look at those. There's three of those. You see each one has a nitrogen with a positive charge on it. So you have to recognize these as basic polar side chains. Finally, going back to the list, the last one is acidic. The R group makes the amino acid acidic. And you see there's two of these, aspartate and glutamate. And these polar charged non-neutral side chains have negative charges. So they're acidic, but they've lost their acidic hydrogens. And that's why the oxygen is negatively charged. So in 19 of the 20 amino acids, the alpha carbon is chiral. So the only amino acid that's not chiral is glycine that you see on the screen. And that's because the alpha carbon has two hydrogens on it, making that carbon achiral, not chiral. But all the other amino acids, at least one carbon in the structure is chiral, and it's the alpha carbon. Because the alpha carbon has a carboxylic group, an amino group, a hydrogen, and some side chain that we just reviewed. And also, because the amino group is on the left side of the structure as it's written, as you see it there, makes them L amino acids versus R. So the amino acids are all L amino acids in living systems. So amino acids exist as zwitterions. It's a dipolar ion, and you see a picture of it here on the right. That's the way the amino acid was presented to you just a few minutes ago, with the amino group having a positive charge, 4 bonds on the nitrogen, and the carboxylic group doesn't have an acidic hydrogen on it. So when you have that, zwitter means 2 in German. So when you have that, you have 2 charges. You have a positive charge and a negative charge. They negate each other, but you have those charges there. And the non-ionized form that you see on the left, where the nitrogen doesn't have the positive charge, the carboxylic group has acidic hydrogen. That doesn't exist. So the amino acids exist in various forms, and one of them is this zwitterion that you're looking at now. So this is something that you have to know about the isoelectric point. So it's the pH of a system in which the amino acid has a net charge of 0. That is, the amino group has a positive charge and the carboxylic group has a negative charge. So that happens at various pHs. So pH at the isoelectric point is around 6, 7. So now the system can have pHs above the isoelectric point or below the isoelectric point. And this you have to know. So if the pH is above the isoelectric point, that's the first sentence there, then that's a basic situation. The amino acid has a net negative value. Why? Under basic conditions, what's going on is the carboxylic group exists without its acidic hydrogen, and the amino group that at the isoelectric point has a positive charge on the nitrogen, now under basic conditions loses one of its hydrogens and it has no charge. Therefore, the net charge on the amino acid is negative. So at pH values below the isoelectric point, that's acidic conditions, the amino acid has a net positive charge. Why? Under acidic conditions, the carboxylic group then gains a hydrogen, so it has no charge. The amino group it keeps its hydrogen and has a positive charge because it has 4 bonds on the nitrogen. And therefore, the whole amino acid has a net positive charge because the positive charge on the nitrogen is not negated by the negative charge on the carboxylic group. The carboxylic group has its acidic hydrogen under acidic conditions. So because of these possibilities, amino acid solutions can act as buffers. When in the presence of acid, then the amino acid can take the acidic hydrogen and put it on the carboxylic group. On the basic conditions, OH, hydroxide, the hydrogen on the nitrogen changes the hydroxide into water. So therefore it acts as a buffer. And we'll be doing a homework problem, a couple of homework problems along these lines. So when the amino acid then is a zwitterion, it exists as a white crystalline solid, relatively high melting point, and of course high water solubility because it has a negative charge, a full negative charge, and a full positive charge, making it very polar, and so water soluble. So one important reaction of amino acids, particularly when they are in one of the components of a protein, is the formation of a disulfide bridge or bond. So you know this reaction from chapter 4 or 14, that two sulfides can be oxidized and form a disulfide bridge or disulfide bond. And you're going to see that this is an important reaction as we go forward in this chapter. So the amino acid that undergoes this reaction is cysteine. It's the only amino acid that has the sulfhydryl groups that undergo this reaction. And again, this sulfur-sulfur bridge, this disulfide bridge, is going to be important as we go forward and look at the structure of proteins. So the reaction that you see on the screen now is the reaction of a carboxylic acid and an amine to make an amide. Now, in chapter, think about it, in chapter 6 or 16, we said this reaction doesn't happen. We said it's just an acid-base reaction and we don't form an amide. We said to form an amide, you need an acid chloride or an acid anhydride and an amine. But in a biochemical reaction, it does happen. So it happens in humans, for example, or in animals, for example. So there's a carboxylic acid and an amine, and it makes an amide. And so this carbon-nitrogen bond on the right in the product, that carbon-nitrogen bond we call an amide linkage. You know that again from chapter 16, but it's also called a peptide linkage because it is the very reaction that we need for amino acids to combine to make peptides, polypeptides larger, and and proteins even larger molecules. So the first significant reaction of this chapter is the reaction again of two amino acids to make a dipeptide. So the two amino acids that are here as an example are the two simplest amino acids: glycine, where the R group on the carbon is a hydrogen, and alanine, where the R group is a methyl group. So you have to know this reaction. Again, it's nothing new. You've seen something like this before. And so when it's the carboxylic group of glycine and the amino group of alanine, then you make one kind of dipeptide that we're going to call glycoalanine. But down below on the screen, if it's the carboxylic group of alanine that reacts rather than the carboxylic group of glycine, if it's the carboxylic group of alanine and the amino group of glycine rather than the amino group of alanine as up above, you also make a dipeptide, but it is a different dipeptide. So if you examine the two dipeptides, one up top, on the carbon, the two hydrogens on it, there's an amino group. The bottom dipeptide, amino group, is on a carbon with a methyl group on it. So looking at the top dipeptide, the carboxylic group is on a carbon with a methyl group, whereas the bottom one, the carboxylic group, is on a carbon with two hydrogens on it. So it's important which end of the amino acid react to form the dipeptides. So peptides are larger than amino acids, but they're a combination, as you just saw, of amino acids. So if you combine more than two amino acids, you get a peptide. If you combine, say, like 50 amino acids in a chain, in a polymer chain, then you make a polypeptide. And if you go larger than that, if you combine more than 50 amino acids, then you make a protein. So the convention is when you write these peptides, polypeptides, and amino acids, the nitrogen, the amino group, is on the left, and the carboxylic group is on the right. So the amino group is called an N-terminal residue, and the carboxylic group is called a carbon C-terminal residue. So what you're looking at on this screen is a tripeptide made up of alanine, glycine, and valine in that specific order. So in this chapter, there's a lot of medical information. I'm not going to review the medical information, and I'm going to let you read that yourself. I'm just going to focus on the biochemistry of the chapters. So what you have here are hormones, and these hormones have disulfide bridges, and you can see that in the two structures that you see. And in each of the balls, in these hormones, each of the balls is a particular amino acid combined in that sequence. And when you combine these amino acids, and the key bond is the amide or peptide bond between a carbon and a nitrogen. These two particular hormones have disulfide bridges as key bonds in their structure also. So I'm going to skip the medical. So, and then talk about proteins themselves. So proteins are very large, naturally occurring polymers. They have huge molecular weights. One molecule can have a weight of 6,000 to several million. They're too large to pass through cell membranes, so they're contained inside normal cells. And if they leak out, then you have a medical problem. Now, protein in the urine indicates a problem with your kidneys. Protein in blood indicates a problem with your heart. So proteins take on the form of zwitterions, again, with the amino group positively charged and the carboxylic group negatively charged. And they have a certain isoelectric point, again, it's pH dependent. Proteins behave as buffers. Again, they can take a basic solution, turn it into a relatively neutral solution, or take an acidic solution and turn it into a relatively neutral solution. So they can be in solution in the form of stable colloidal dispersions. That is, they form micelles, such as you've seen in the chapter we did on lipids and fats. And they the repulsion forces are weakest at the isoelectric point. So what you want to have when, when you have these protein micelles is you want to have net charges on the protein. You don't want to have a protein with no net charge. As you see here, when that happens, the proteins clump and precipitate from solution. You want to have it that the net charge in the protein is either positive or negative, so they repulse each other. The micelles don't come together and clump. We're going to, again, skip all the biomedical. So some proteins exist as long, rod-shaped or string-like molecules. They're fibrous proteins. The globular proteins are spherical. As you see in the picture, the positive and negative charges of this particular protein are on the outside, and all the carbons and hydrogens are on the inside. That's the hydrophobic region. The charged components of the protein are all on the outside in contact with an aqueous system. So simple proteins contain only amino acid residues. But conjugated proteins have not only the amino acids, but they also have prosthetic groups. And so what you see here, this picture, is a hemi. And it's a key component to hemoglobin. You see that the picture on the left is hemoglobin, and it's got 4 hemi groups in it. If you can look at the structure, and you see there's 4 of these structures with iron in the middle of it. So these groups then that are part of a conjugated protein are called prosthetic groups. So proteins have 4 kinds of structures. The first is the primary structure. So the primary structure of a protein is the sequence of amino acids, a particular sequence. And again, what bonds one amino acid to the next is that carbon-nitrogen bond that you see in the protein at the bottom of the screen. Again, we call it either an amide linkage or a peptide linkage. But that's the key bond. And so it is the order of the amino acids in the protein that gives the protein its particular characteristic. There's also secondary, tertiary, even quaternary structures that a protein can have. So this you have to know. So let's go ahead and look at the secondary structure. So the secondary structure of a protein is determined by hydrogen bonding. And it's a hydrogen bond between the oxygen and the carboxylic group and the hydrogen that's on the amino group. So proteins can exist in an alpha helix form and or a beta pleated sheet. So better to see on the next screen these structures. So here you see the alpha helix, and I think the structure on the right, they're both the same, but they're just depicting it differently. I think it's easier to look at the structure on the right, and there you see that the hydrogen on a nitrogen is hydrogen bonded to the oxygen of a carboxylic group. Okay, they both should show that, but I think the one on the right, the alpha helix on the right, better depicts that kind of hydrogen bonding. So that's the alpha helix. The beta pleated sheet, again, it's a different form, but it's the same kind of hydrogen bonding. The big ball is the oxygen, and the little ball is the hydrogen. And you see the dots indicating the hydrogen bonding. And that's what keeps the alpha helix and beta pleated sheets in the form that they're in is that hydrogen bonding. So that's the secondary structure of a protein. Proteins also have a tertiary structure. So here you see a particular protein, and it has a coil structure. It has a beta, part of it is in the form of a beta pleated sheet, and part of it is in the form of an alpha helix. So all that can exist in one protein molecule. But the tertiary structure, there's four ways a protein can exist in a tertiary structure. And it's a 3D shape of the protein. It has to do with the R groups. Up until now, it was only the skeletal structure of the protein that we were speaking about. The amide bonds, the hydrogen bonding between the amino hydrogen and the carboxylate oxygen. Now we're speaking about a tertiary structure of a protein that's defined by interactions of the R groups that hang off the protein molecule. So let's go through each of these in this photo that you see here. If the protein has cysteine as one of its amino acids, cysteines can react to cysteines in the same polymer and form a disulfide bridge. And then again, that helps to define the structure of that particular protein. Salt bridges can exist also. So if the protein has a base, a charged base amino acid and a charged acid amino acid, and the negative and positive charges can attract each other and form a salt bridge. So the example here is aspartame and lysine. There's a third kind of interaction, and the example is two hydrophobic interactions, and you see phenylalanine, and it has a aromatic ring in its amino acid, and the interaction of these two aromatic rings form a hydrophobic interaction. Very weak, but still a hydrophobic interaction, again, defines the structure of the protein. And finally, you can have hydrogen bonding, not the kind of hydrogen bonding between the amino group and the carboxylate group, which is in the secondary structure of the protein, but in the tertiary structure of a protein, you can have hydrogen bonding between the R groups. And in this case, it's serine. It has a R group of CH2OH, and those two CH2OH groups can interact and create a hydrogen bond. So you saw this picture before. In an aqueous environment, the protein can form some kind of a micelle, where you have a hydrophobic region of carbons and hydrogens in the middle, and all the charged and polar R groups can be on the outside. So all proteins have primary, secondary, tertiary structures. Some have even quaternary structure. And the quaternary structure is the arrangement of the primary, secondary, and tertiary structures. That particular arrangement would be the quaternary structure of the protein. So conjugated proteins can contain a prosthetic group. We saw an example of that earlier. It's the same one here. It's the hemi group that, again, is part of hemoglobin. So, again, a picture of hemoglobin, and you see the four prosthetic groups in it, the four hemi groups in the picture. And hydrophobic forces hold these subunits together. That's the quaternary structure of that particular hemoglobin protein. So proteins can undergo reactions. And again, you know this reaction It's the hydrolysis or the saponification of an amide. A protein is a polyamide. And so under acidic conditions or basic conditions, then the amide bond, the peptide bond, is cleaved, making smaller peptides, and even if it continues, amino acids. If when a human digests a protein, then the enzymes are catalyzing this same reaction rather than acids or bases. And proteins can be denatured, and they lose biological activity when that happens. And there's various ways that proteins can be denatured. So a common way is heat. That's what happens when you break an egg and put it in a frying pan. The clear part of the egg, which is the protein part, becomes white because the heat in cooking denatures the protein. But it can happen with ultraviolet light. And also, there's other involvements the protein can have, which leads to denaturization of the protein, be it organic solvents, strong bases, detergents. We're going to come to heavy metal ions in the next chapter on enzymes, but that denatures and makes the protein inactive. So that's the end of chapter 19. So give me a minute, and I'm going to then hopefully get up the next chapter, chapter 20. Okay, so hopefully everybody now can see chapter 20. If you can't, let me know. But hopefully everybody sees the picture for chapter 20, enzymes. So the way I teach this chapter, I do skip over some parts of it. So we're going to do that. An enzyme is a catalyst, like acid catalysts, like catalysts like the hydrogenation reactions that we learned earlier. They involve a platinum or a palladium catalyst. So enzyme is a naturally occurring catalyst. And what it does is it lowers the activation energy in a reaction. And that's the picture that you see in front of you. Is you see the reactant at a certain energy level, and then you see a pretty high activation energy, and then you see the product lower in energy than the reactant. So this is an exothermic reaction. And you see that uncatalyzed, it's a high activation energy. And then when it's catalyzed by an enzyme, the activation energy is very low. So what that means is speed. If a reaction is uncatalyzed, typically it's going slower than if it is catalyzed. So it's the speed of reaction that's important. So I got to do something here because I've got a window that I have to get rid of. So give me a minute to do that. Control, Alt, Shift. Okay, all set. So some enzymes have absolute specificity, like the one you see at the bottom of the screen. Urease catalyzes only the hydrolysis of urea. And an enzyme can have relative specificity, where it acts only on structurally related substances like lipase. Remember in the chapter on fats and oils, we said you can hydrolyze a fat or an oil with either acid catalyst or an enzyme called lipase. And so that's an example of a relative specificity, acting only on structurally related, in that case, on fats and oils. But also, some enzymes react stereochemically. We talked about this before, that the human body can recognize one enantiomer, one chiral molecule versus another, the opposite, right? They're called enantiomers. So that's an example of stereochemical specificity. So enzyme activity can be regulated, and it's generally regulated by cells. If the cells don't regulate the the reactivity of enzymes, the enzymes destroy the cell. The reactions become out of control. So we're not going to focus on naming enzymes. We're going to skip that part. So you're not going to be responsible for naming a particular enzyme. So it's going to skip all the naming. So some enzymes require a second substance to be present, called a cofactor. And there's two kinds of cofactors. If the cofactor is an organic molecule, it's called a coenzyme. And if it's a inorganic, like a vitamin or a metal like calcium, for example, then it's just called a cofactor. And there's a list. And you certainly don't have to memorize the list of vitamins and the coenzyme forms and what they do. I'm just going to skip over that biomedical portion. So what you want an enzyme to do is to find a substrate. Like in the example before, the substrate was urea. So you want urease, the enzyme, to find urea and form an enzyme substrate complex. That's key. And that reaction is reversible, as you see. And then that enzyme substrate complex can decompose, produce a product, and regenerate the enzyme. Right? A true catalyst or an enzyme has to be regenerated. It can't be part of the product or it loses its function. So here's a real example. So many enzymes are named after the substrate that they react with. So sucrase is an enzyme that reacts with sucrose, making a sucrose-sucrase complex. It yields to sucrose, it yields to glucose and fructose, basically cleaves the sucrose, and again regenerates itself so it can act on other sucrose substrates. So how does it find this? How does it make this enzyme substrate complex? One method is called the lock and key model. That is, the enzyme again is a part of a protein. So the enzyme is a large molecule, and the substrate generally are much smaller molecules. That's why in the picture that you see, the enzyme is large and the substrate is much smaller. So in some reactions, the substrate perfectly fits into an opening that the enzyme has and makes this complex, this enzyme substrate complex. So that's called the lock and key model. So that's one model. But another one is that the enzyme, before it bumps into the substrate, has an opening, but it's the wrong size. But as the enzyme gets closer to the substrate, it changes its structure, and it accommodates the enzyme. So that's the induced fit theory. And it only happens when the substrate gets close to the enzyme. We're going to skip over enzyme activity and go to these four charts. So this you do have to know. So when you have a lot of enzyme present, the rate of reaction continues to increase as long as you have an increase in enzyme concentration. So the chart looks like the one that you see on the screen. So that's one situation. The second is you give a rate increase when you're increasing the amount of substrate. So it begins to look like the previous chart where more enzyme yields a faster and faster rate of reaction. In this one, though, the rate of reaction tails off at some velocity and doesn't increase, stays at a high level, but doesn't increase any longer. And that's because you run out of enzymes. You have a lot of substrate, but not enough enzymes to catalyze all the substrate that needs to react. So you have a tailing off of the rate. Again, at a high level, but the rate increase stops. Third situation is enzymes only work at certain temperatures. So you have a temperature at which the enzyme activity is optimum, and above and below that temperature, it's not. So we saw before that proteins can be denatured by heat. So on the high temperature side of that chart, you're denaturing the enzyme because, again, an enzyme is only a portion of a protein. And pH. Again, we saw before with proteins that you can denature a protein just as you can make an enzyme not react at all by changing the pH. So the best pH is around 7, but in the gut, for example, in the gut of a human, it's very acidic. So enzymes that work there for digestion typically work on the acid side of pH 7. And then there's some enzymes that work best on the base side of pH 7. And so you're seeing that here in this chart. Again, in intestines, the pH is very, very low, very, very acidic. But in other areas, the pH is greater, like in the intestine. So inhibition. So enzymes can be inhibited two different ways. Either you have irreversible inhibitors or you have reversible inhibitors. So an example of an irreversible inhibitor are poisons. So cyanide poisoning, mercury poisoning, lead poisoning, these are all generally irreversible. So what happens with cyanide is that cyanide, when it's taken as a poison, complexes with cytochrome oxidase and respiration stops. So death in minutes, you can see in old movies, old wartime movies, that some people take these cyanide capsules to commit suicide. So that's what happens. It interferes with respiration. And there's an antidote for cyanide poisoning, and it's a chemical called hypo. It's an old chemical that photographers used to use to develop photographs. And you see, hypo, I think it's sodium thiosulfate, and it interferes with the cyanide complexes with the cyanide and renders it unusable. So heavy metal toxicity, here's where cystine takes a role. If you have an enzyme that has cystine in it, and many do, the cystine again has these sulfide groups on it, and mercury or lead, in a reaction again that we saw in chapter 14, can preferentially react with the sulfur and cause the enzyme not to function as it should. Again, it's the preferential reaction. The enzyme prefers to react with the mercury, in this case mercury cation, rather than reacting, for example, with a substrate. So they talk about chelating and what to do if you have lead or mercury poisoning. We're going to skip over that. So enzyme inhibition. Now we're going to talk about reversible. Before we talked with cyanide and mercury and lead, it was irreversible. Now this is reversible. So we're going to spend a couple of minutes looking at the reaction in the upper right-hand corner of your screen. So one type of reversible inhibition is where the inhibitor fits in the opening that the enzyme has preferentially in place of the substrate. So what do you do in that situation? So to make the reaction reversible, you increase the concentration of the substrate. So that's depicted in the two reactions in the upper right-hand corner. This is Le Chatelier at work. So what reaction you want to see is the top one, equilibrium 1, where you make an enzyme substrate complex. What you don't want to do, what you don't want to see is the enzyme inhibitor complex. So by increasing the concentration of the substrate, you drive the top reaction to the right. But to go to the right, you not only need substrate, you need enzyme. So as you pull enzyme out and make the enzyme substrate complex, you need more and more enzyme. And so that has the effect of reversing the second reaction to produce enzyme so that the enzyme can be used in the first reaction. So that's the effect of increasing the concentration of substrate when you have reversible enzyme inhibition. And here, this is a competitive situation. Again, it's inhibition, and it's the role of sulfonamide that can have in curing an infection. So the sulfonamide looks a little bit like the para-aminobenzoic acid that you see at the bottom of the screen. You see they both have aromatic rings. There's an amino group on the aromatic ring. But in the case of the sulfonamide, it's a sulfur that's there. In the case of the para-aminobenzoic acid, it's a carbon, of course. But they look similar. And so the sulfonamide substitutes for the para-aminobenzoic acid. So folic acid, which bacteria needs to multiply, isn't made. Because in place of it, you make the similar compound, but made from sulfonamide, and bacteria can't grow. So a non-competitive inhibitor then binds to an enzyme not at the reaction site where a substrate would go, somewhere else on the enzyme, and causes the structure, the stereochemical structure of the enzyme to change such that the substrate no longer fits on the enzyme and the reaction doesn't occur. So zymogens then are inactive precursors to enzymes, and they're released when needed, released by the cells when needed. So allosteric regulation of an enzyme is one way the cell can regulate the reactivity of an enzyme. So you can get modulators that, again, bond to an enzyme and cause its structure to change such that it's either going to reduce the reaction with a substrate, or perhaps maybe even increase it. But that's called allosteric modulation, changing a substance that will change the structure, the stereochemical structure of an enzyme. Okay. I think that's it for the chapter. We're going to skip all the rest of the chapter. So I gave you the homework assignment for chapters 19 and 20. During the week, I'll try to do most of the problems and send them out to you. Once again, for those of you that joined late then, the last class is next Thursday. That's the last class of the semester. That's when you take exam 3 on chapters 17, 18, 19, 20. I'll correct them pretty quickly and get the results back to you. And then I'll begin sending you emails about the final exam. Any questions on, again, on what I covered this morning, please send me an email and I'll try to answer pretty promptly. So have a good rest of the day. Take care. Bye-bye.